addicted.
Yeah, so thanks to the organizers and also for you staying here, because I know the next thing after me is lunch. So at least the good thing, hopefully, is that as I'm going to talk about taste. So if you think about the taste of medicine, what kind of taste do you think about?
Bitter.
Bitter. Yes, so this is the most common taste of therapeutic drugs, noted early on, even earlier than that. But actually, it is a major problem specifically for pediatric drugs because, as was mentioned in the previous talk, when there is no compliance, when it's so disgusting that the children will not take it, it doesn't matter how good the drug was, it's not going to work. Sorry. So it is important to know, this is one of the reasons why it's important to know the taste of a drug. So taste recognition is usually presented in this way. It's important for survival. There are only a few basic taste modalities. Some of these taste modalities, and this is, for example, different from odors, where you have numerous types of odors. Here we have just a few basic taste modalities, with many of them recognized by GPCRs, G protein-coupled receptors, including the sweet taste, for example. And here I'm showing not only a cartoon of the receptors that are responsible for recognizing these taste modalities, but also the innate response of newborn children or animals. And you can see the licking response, which is an innate response that's a positive response. Animals are attracted to to sweet taste, and this is thought to be useful for detecting calorie-rich foods. And on the other hand, a gaping or kind of towards throwing up reaction of newborns towards bitter compounds. And this is thought to be useful for detecting poisons and not injecting them and then not dying from them. What I'll try to do during this talk is maybe to question a little bit this paradigm. But it's important to note that that's what we're always saying. In the beginning of every paper we say or read, bitter taste is a marker of toxicity, and then we continue. And there are many bitter compounds. And when we started to work on this, we wanted to know which compounds actually have a bitter taste. And INI in my lab has established the BitterDB, a database of compounds that were either mentioned to be bitter in Merck index, because people back then, not any longer, but people used to taste what they have synthesized. So there's still records in the Merck index about taste of molecules. In Finaruli Handbook of Flavors, and then because the taste receptors are known, it is also possible to test in cell-based assays to see which compounds are activating particular receptors, and then to know that these are also bitter ligands. So there are over 700 different compounds. They are very different in terms of chemical structure and physicochemical properties, and they are recognized, as I mentioned before, by G protein-coupled receptors. The largest family of GPCRs are the olfactory receptors, but bitter taste receptors are a pretty big family as well. Not in any animal. In humans, there are 25 different subtypes for recognizing bitter taste. In rodents, there are a similar number. In chicken, for example, there are only 3, so there are many questions about evolution and so on. But for us, it's important to know these receptors, because now actually there are crystal structures for over 40 different members of the GPCR superfamily. Unfortunately, none of them for the bitter taste receptors. Okay, so I said there are 25 different bitter taste receptors, but even if we look at ligands of a single bitter taste receptor, there is a huge variation in the chemical structure of the ligands of that same receptor. And this is shown here by Tanimoto coefficient that is very low, close to 0. There is one family that actually presents homolones that come from hop, from beer. So it's important. It's bitter. And there is a family that has chemical similarity, but there are others that are very dissimilar. So we want to, given all this diversity and complexity, we still want to be able to predict bitterness of a molecule based on its structure. So there are different ways to do it. First of all, we can forget about structure and just use human knowledge and do data mining. Unfortunately, it cannot be fully automated because often you would get, anyway, sucrose is often mentioned in an experiment that tested different things, including this. So anyway, so you have to add manual labor here also to the automated searches. Then we can use we can search for ligands that are similar to known particular bitter ligand, and we have done that. You can look for ligands that fit into a 3D model of receptors, similar to what it did showed you before. We have done that as well, but we have also found that the same binding site can actually recognize different ligands in different ways. So if I'm checking if a particular molecule would fit into a particular binding site, and I see that it doesn't fit, it still doesn't mean that it cannot activate it. So if it does fit, okay, there's a better chance that it would activate than taking a random molecule. But if it does not fit, it doesn't tell me anything actually, because it could be different way of binding, or it could be actually different receptor out of the 25 that we have. So what can we do if we want to have a general idea about bitterness, or if I want to take a molecule, look at it and say, okay, it's likely to be bitter, it's likely to be non-bitter. So of course we go to machine learning. And for machine learning, the major thing is, of course, to have good input data. So we have the bitter DB, so we have the true positives, the bitter compounds. Here we also had to establish non-bitter data set. So in principle, you could say, okay, take any random molecule, that should be the non-bitter for you, but I'll show you soon that that's actually not so true. What we did was, what Ayana did was, look here, you can see the random molecules represented in yellow on a principal components axis. The non-bitter flavors are in red, so you see that actually it's kind of a concise kind of chemical space. And then the bitter in green are all over. So it was really important for this work. We've been working on this for a while. It was really important to have very good quality non-bitter set, and that was taken from sweet but not bitter, right? Not like artificial sweeteners, because they are both sweet and bitter. So sweet, which are not bitter, tasteless, and so on. And also the bitter. Then the descriptors that we used in this case were physicochemical descriptors of the molecules, as well as admitox descriptors that were developed for pharma for predicting toxicity of compounds, because of this association that we knew about, which is the paradigm that bitterness signals toxicity. And then the machine learning method that Ayana used after some preliminary testing was AdaBoost, which is basically an adaptive decision forest method. So there are many decision trees, and it's enough for each one of them to be just a little bit better than random prediction. Together, it's like wisdom of the crowd. Together, it gives quite a good prediction. What is quite a good prediction? So here you see the training set, but of course more important is how it does on holdout set. And we see that on holdout set, we're about 80% sensitivity, specificity, and accuracy. And then of course we need to repeat it for external data sets that were not used, not seen by us at all. And we did it with external sets that were published later, with sensory human panel, which we did ourselves. So we predicted some molecules bitterness, or in this case non-bitterness, and then tested in sensory panel. And then we used data mining on compounds for which taste should have been or might have been known, and we looked at co-occurrence with, actually manually looked again at their being mentioned as bitter or not. And so all of these tests assure that indeed we are doing pretty good. So about 80%, give or take, accuracy. There is no one real, because it's not a simple model. I cannot take one physicochemical property, say this is what going to tell you if it's bitter or not, but total charge contributes probably the most data, even though we have to have the different components and the different descriptors. All right, so now we have this predictor, and now we can look at different data sets and see how frequent actually do we find their bitter compounds. And we looked at food DB, right? We want to know actually what's going on in food. And our prediction, or intuitively, we would think, well, not so many bitter compounds there. In drug bank, as you told me before, this is a typical taste of drugs, so we would expect many bitter compounds in the drug bank. CHUJI, as a representative of random molecules, again, naively, we would think not so many bitter ones. And natural products, well, open question. So what we found was that there are about 40% compounds with some bitterness in the food DB, higher than 66% in drug bank. Random, it's not as high as in drugs, but still actually not negligible. So going back to from where I started, actually it's not a good idea, even though it has been done in the past, to assume that a random molecule is tasteless or non-bitter. Actually, it has a pretty good chance of having some bitterness, and natural products are very rich in bitter compounds. So what we find here is that some bitterness is quite often encountered, both in food and in random compounds, that natural products and drugs are very much enriched in bitter compounds. And of course, it would be really nice to add to all of this intensity, bitterness intensity, how bitter that is. Is it strongly bitter or weakly bitter? Currently, there is not enough data in our hands, at least, to, or probably anywhere, to have develop a predictor, because the data on intensity is very scarce. So just to convince you that it is really so that in food we do encounter bitter compounds, here are some names of bitter compounds, and here are the foods that have these compounds. Now, going back to the question about toxicity and bitterness, so these foods are actually, many of them are quite good and even maybe healthy. So then we wanted to check whether this paradigm that we start with in almost every paper, bitter taste signals toxicity, actually how well is it grounded in facts and what we know so far. So Ido, he's also in the audience, studied this issue. So we had the bitter DB, we know what is bitter. Then Ido compiled a large data set of toxic compounds from the World Health Organization, and then a smaller data set of compounds that have oral acute toxicity, right? Because the whole idea is that it's supposed to do something with oral and acute, and that's how the bitter taste should protect us from consuming these poisons. Okay, so there were only 10 compounds that were common to the bitter DB and to the focus toxic data set, but that's maybe because we don't know the taste of some of the compounds. But now we have the predictor, the bitter predict. And so indeed, in the Combitox, around 60% are predicted to be bitter, a little below, and a little higher than that predicted to be bitter in the focus toxic library. So it does seem to have bitter taste, but again, even less than in drugs, or similar to what we have in drugs. And still, there are many poisons that are not known or predicted to have bitter taste. So that means that if we are going to rely on bitter taste to save ourselves from poisons, it shouldn't be the only thing you're doing, right? Because it can be tasteless, like arsenic, for example, and still be toxic. And so there are like 40% of toxins like that. But maybe it's something to do still with ratios and the concentrations, right? Maybe if we look at bitter compounds, they will have LD50 values, so the lethal doses that are needing to kill half of the population that was exposed to that compound. Maybe the LD50 values will be lower. So if I'm looking at a data set of non-bitter compounds and I have a distribution of LD50 values shown here, so this is fatal upon a few drops. And the other one is maybe harmful or non-toxic, so basically non-toxic for us. And there are others for which no LD50 are knowns, but if you're looking at these known ones, this is the situation for non-bitter. Before we did this analysis, I was expecting, and based on what we know, I was expecting that within the bitter group, we will have a lot of these guys here, toxic, fatal and toxic, and less of the others. However, that's the picture we get. So basically the same trend, maybe some specific difference, but basically the same trend. Not so many fatal, most of them harmful or hardly harmful. So 60% of bitter compounds have some level of known toxicity, but distribution of oral LD50 values for bitter is kind of similar to non-bitter. So again, not a very strong indicator of toxicity. Then we checked another hypothesis, which is saying the following. Remember, I told you that there are 25 different subtypes of bitter taste receptors. And it's not really clear, actually, why we have so many subtypes. So there was one hypothesis that said this is for backup. So if we have something that's very dangerous, then perhaps we would have more than one bitter taste receptor that is activated by these dangerous compounds, so we have a backup system. So we thought that perhaps we'll find a negative correlation between the LD50 and the number of receptors that are activated. So we have something very toxic, many receptors react to it, have something that's not toxic, one is enough, but we didn't find such a correlation. Okay, so the number of bitter taste receptors does not correlate with the LD50 values. All right, so we can summarize what we've found so far. We have the BitterDB, which is a database that is it's free, it can be browsed, sorted, searched, and uploaded, which is really important because if you know of compounds that are bitter or toxic or non-bitter, we really like to tell us more if you don't find it already in the database. We have developed the bitter predict classifier, and despite the really big challenge because of the tremendous chemical diversity of the bitter compounds, that was nevertheless possible. And this is now useful for example, for flagging of drug candidates for bitterness. So in the early stages of drug development, and this is something that I learned actually from a pediatric formulation initiative, they actually have assay. Currently what they use is a brief access assay by rodents to see if rodents are drinking or not drinking the solution with the compound as a very early indicator of whether that compound is going to be aversive or not. So what we are now offering is something that is costless, basically, because it's electronic, and can predict if it's going to be bitter, and can, of course, be improved because this is a system where the more data you get, of course, the better your predictor is going to be. To flag these compounds, this does not mean that we don't want to develop them into drugs. Not at all. As we saw before, majority of the drugs do have bitter taste, but this is something to consider when considering in the future if it's going to be suitable for children, if there are going to be problems with formulation, and so on. So this, as a flagging, it can be useful. And then we discovered that actually this suggested connection between bitterness and toxicity is actually maybe not as strong as we have thought. And I'm even starting to think that maybe detecting bitterness, even though usually it is said as being a marker for toxicity and something to warn you against consuming poisons, there is another side to it, because actually there's also self-medication for animals, and actually in ethnographic medicines, where bitter taste is actually considered something that might indicate therapeutically useful substance. So, okay, so maybe we don't always need to be so scared from bitter taste. Maybe there are also some good things in it. And an additional very interesting point is that the bitter taste receptors have been found in many different tissues. So they're expressed in the lung, in the heart, in the thyroid gland. So they must have additional roles. They must have roles that are not related to oral detection of poisons. And what is also very interesting is because existing drugs are already bitter and already drugs, it might be a really good opportunity for repurposing bitter drugs, but for novel indication, now working via activation of bitter taste receptors. And the indication that is most suggested for that is asthma. So bitter tasting compounds were shown to cause bronchodilation, and this is one of the suggested directions for novel anti-asthma drugs. So I'll just go back to the first slide and say the following, that even though we usually say, okay, we have the attractive and the good and the nutritious, sweet tasting compounds, and we have the dangerous, the bitter, the toxic, the poisonous compounds. I think that actually maybe we need to think a little bit this issue over for two reasons. One is because in the modern diet, actually attraction to sweet taste is not as it used to be for hunters-gatherers. So actually this is becoming maybe a dangerous thing, the fact that we are so much attracted to sugar. And then on the other hand, the bitter compounds, it's true that some of them are toxic, but also some non-bitter are toxic, and also some bitter are non-toxic. So this relation is not so strong, and so maybe even though innately newborns and little children are aversive to bitter taste, maybe it's something that we should consider as something that we as healthy eaters should get used to and have healthier diets. But it's a question, not proven yet, but it's just food for thought. And I would like to acknowledge people who have contributed to this work and to thank you. And I'll be happy for questions, but I'll understand if you just prefer to go eat. Thank you. Thank you.
Personal experience, I would say that liking bitter taste is something that is acquired. Is there any, do you look at that kind of biological evidence of?
Yeah. So two ways to answer. First of all, it's true. And there is a researcher, Julie Manella, and she's called Julie Manella, and she's at Monell Center for Chemical Senses, and she's actually working on changing diet from very early on, including breastfeeding, and even during when it's still a fetus, what the mother eats, and so on. And so she sees that exposure multiple times. It's like 20 times early on can change acceptance and preference. I think there's also a physiological reason, or I don't know, evolutionary sense to that, because it's not a error-proof system, right? If it's bitter, maybe it's poisonous, but maybe it's not. So if you have eaten it and nothing happened, actually it makes sense that at some point, right, you will not neglect that food that actually could be useful just because it might have been. So if it happened, and this is why also early stages and through mother, if it's mediated, then it's an acquired taste because nothing happened, it went well, and so then at some point it becomes something that's acceptable. How it works on psychologically, neurologically, it's not so clear.
What's the affinity of these receptors?
Yeah, it's low. It's micromolar typically, and there is also difference between. But it makes. I mean, you don't need this more than that for detecting something. Usually for eating. Yeah, if you have. Yeah, like usually. And there is also a difference between in vitro and in vivo. We try to test it actually in chicken because they're just 3 receptors, so it was like a clean system to see. So there can be difference of up to 100, so 2 orders of magnitude higher in vivo thresholds than in vitro. So in vitro it's micromolar, in vivo it might be even lower or the same, or like, and in some cases, we have some very few examples of submicromolar affinity, and actually we're hoping to find more because maybe in the extraoral tissues, when the role of these receptors is different, but we are looking but have not found yet nanomolar ones.
Combining the fact that you say that there's these receptors in different tissues, and the fact that we do see enrichment in after products. Could it be a different story that you already had these receptors, and in fact it's sort of an advantage to develop this don't eat me taste?
So the idea is basically about this poisoning is that the plants are trying to protect themselves from being destroyed by producing these secondary metabolites. Some of them are poisonous and toxic, and some of them and bitter, and some of them are just bitter, but that has become enough because the herbivores have developed the ability to taste bitterness and to avoid it. So actually at this point it's enough just to be bitter, and you don't really have also to be toxic because you won't be eaten. So the reason we wanted to look at natural products is because the evolution cannot cope with what was developed by synthetic chemists in the last century. So actually it's not fair to look at CHUJI for compounds, and actually we have to concentrate on naturally occurring ones. And indeed, I agree with you, it's expected that it will have a high percentage of bitter compounds because that's the idea, that's like the protection mechanism. But we didn't.
Maybe the initial purpose, even though we see it also in olive tissue, that there is a different purpose and it's a sort of hijacked.
Maybe, yeah. For the plants, of course, it might have had other roles, yeah.
Thank you very much.
Thank you.